Dear participants, please wait 10 minutes, then we will start today's session. Good evening, everyone. A very warm welcome to all the respected dignitaries, faculty members, researchers, professionals, scholars, and their participants joining us from different parts of India and abroad. On behalf of Edu-Mentors Pvt Limited, it is my pleasure to welcome you to all to the 21 Days International Summer School Training Program on Climate Resilient Agriculture and Allied Sciences. This international training program has been thoughtfully designed to provide participants with practical knowledge, emerging technologies, and scientific advancements in agriculture and allied sciences through expert lectures delivered by eminent academicians and scientists from renowned institutions. Before we begin today's technical session, it is my privilege to introduce the visionary behind this initiative, Mr. Ashutosh Behra is the Chief Executive Officer, CEO of Edu-Mentors Pvt Limited and the Organizing Secretary of this International Summer School Training Program. Under his leadership, Edu-Mentors have been actively organizing international conferences, faculty development programs, training workshops, book publication initiatives, and skill development programs aimed at bridging the gap between academic knowledge and industry requirements. His vision is to create a global platform where students, researchers, academicians, and professionals can learn from leading experts, collaborate, and contribute towards sustainable agriculture development and innovation. So, we sincerely appreciate your dedication and efforts in making this program possible. Now, it is my honor to introduce our esteemed source person for today's session. We are privileged to have Dr. Ashutosh Sharma, Associate Professor, Faculty of Agriculture Sciences, DAV University, Jalandhar, as our distinguished speaker. Dr. Ashutosh Sharma is currently working as an Associate Professor at the Faculty of Agriculture Sciences, DAV University, Jalandhar, India, specializing in the field of plant pathology, plant microbe interactions, and plant stress biology. With over 14 years of experience, he has authored 150 plus publications. He has 8 edited books and 2 authored books to his credit. He is the recipient of the Punjab Academy of Sciences Young Scientist Award for developing a sensitive detection system for Alternaria alternata. He has secured major research grants from DST, SERP, and CSIR. He has contributed nucleotide sequences to GenBank and microbial strains to the Microbial Type Culture Collection, MTCC. He has 4 design patents and has supervised 15 masters and 4 PhD students. We are truly honored to have such an accomplished academician with us today. I am confident that today's session will be highly informative, inspiring, and beneficial for all participants. Without taking much more time of your valuable, I respectfully invited Dr. Ashutosh Sharma to deliver today's lecture. Sir, the virtual platform is all yours. Welcome and thank you for joining us.
Thank you so much for a wonderful introduction. Just a minute, I will mute my audio from mobile device. Yes, are you able to hear me now? Okay. Slides are also visible.
The slides not visible. सर आपका माइक बंद है. सर आपका माइक बंद है.
Yes. Am I audible now?
Yes, sir.
So I will full screen the PPT. So they are displaying in full screen mode now. So I welcome all the participants on this program, and today we are going to discuss about sustainable mushroom farming. I hope I am audible now and slides are properly visible. Are they?
Yes, sir.
Okay. Mushroom farming, when we listen about mushroom farming, so we know a little bit of what are mushrooms and what is mushroom farming, but doing it in a sustainable manner, in environment friendly manner, using minimum resources and mostly utilizing agricultural waste is important. So before I proceed further, let's start with this term mushroom. What is a mushroom exactly? So many times we heard of this term mushroom. We saw mushrooms in rainy season coming out of the soil. Sometimes we saw some mushrooms on tree trunks, sometimes on the forest floor. So what exactly they are? They are basically the fruiting bodies of fungus. They can be of various colors, various textures. They may grow at uh, in a forest, on a wood log, or we can grow them in a laboratory. So they are above ground. They are macroscopic bodies, sometimes colored or white, and they produce spores. They produce some structures which are meant for dispersal, which are meant for next generation of fungus. So some of them are edible. All the mushrooms which are edible, which are not poisonous, they can be utilized as a food source by human beings. This is a structure of mature button mushroom. Mushroom is a structure. It has gills, it has a cap, pilus, annulus, stripe. These are different parts of mushroom. But we need to understand that mushrooms, they are necessarily meant to spread the spores. Spores are seed-like structures, although they are different from plant seeds, but they are meant for dispersal. They spread to different places and they give rise to new fungal hyphae. Mushrooms are heterotrophic in nutrition. Like plants, they cannot make their own food by photosynthesis. That's why they are usually not green. They can be edible or poisonous. Poisonous mushrooms, they are very, very dangerous if they are taken in, they are eaten. Uh, there is a case, Julius Caesar, that emperor was killed because he consumed the the soup of a poisonous mushroom. So there are so many historic evidences of mushroom poisoning. So we need to understand that all mushrooms are not edible. In this presentation, we will discuss only about edible mushrooms, and that too mostly about button mushroom. So I was discussing about spores. I was saying that the mushrooms, they produce spores, and spores they spread to new places and give rise to new fungal bodies, new mushrooms, right? So they are very close to plant seeds. They have analogy with plant seeds. In this slide, I have kept some chickpea seeds. They are growing, they are giving out their root. Likewise, on the other side, there is a fungal spore which is germinating out with the help of a germ tube. So they are analogous structures, but spore is a simple cell, fungal cell, which can give rise to a new fungal hyphae. Whereas the seeds, they usually have embryo. And embryo is made by sexual reproduction. Using a pollen and an embryo sac, the embryo is made, right? So seeds, they have mature embryo in them, whereas spore can be any vegetative cell giving rise to a new fungal body in case of a fungal spores, right? So mushrooms, they are made by the fungi to spread the spores, to make spores and spread them. There are different types of mushrooms. In this slide, I have kept some mushrooms which are edible. First one is uh, Kida jadi, cordyceps. There's a caterpillar, and a fungal structure is coming out of the head of that caterpillar. This is caused by a fungus called as uh, cordyceps. So caterpillar has a body emerging out of his head. This is mushroom, and this is consumed by so many sports persons and fitness enthusiasts. This contains some essential nutrients, some important minerals, some vitamins. They enhance stamina in sports persons, especially. So this is very famous among sports persons, especially in Asia Pacific region. Then the second one is Gucci mushroom. This is Morshella. This is now it is being cultivated, but earlier it was collected from wild only. This is Morshella, very delicate and delicious fungi, known for its taste. It is usually 3,000 to 4,000 per kg. The third one, this is Rugada. This is a mushroom, small ball-like mushrooms collected from the soil uh, near the rainy season, especially in Jharkhand, Chhattisgarh region. Next one is the oyster mushroom. We also cultivate these mushrooms in our laboratory. Next one is kukdi mushroom. This grows in wild, so people collect them, they make them, and they say that this is like it cooks like its properties, like mouthfeel is just like chicken. So the next slide is of button mushrooms. All of us know about button mushroom. Whatever mushroom we generally purchase from market, generally all of them, they are button mushrooms. Very rarely other mushrooms are sold in the markets. The last one is paddy straw mushroom. This is a very good mushroom grown on parali, paddy straw. So we need to understand that mushrooms are not plants. They cannot photosynthesize. They are not green. They don't have chlorophyll. But mushrooms, they may look like a plant because they have a stem-like structure, some like leaf-like. That cap is there, and they have rhizoids, the thin filaments of fungi growing into the substratum. So they may look like a plant, but it is not a plant. Otherwise, mushroom is more closer to animals also. Why? Because it cannot photosynthesize, but it respires, it releases carbon dioxide. That's why all the culture rooms of mushrooms, where we grow them in the lab conditions, in the artificial conditions, we keep them ventilated so that excess carbon dioxide is not accumulated, which may be toxic for mushrooms. So we are now switching to the next stage of the presentation, how we cultivate mushrooms. So mostly I'll be talking about white button mushrooms. White button mushroom is basically, it is a species of fungi which is saprophytic in nature. This is mostly Agaricus bisporus. It is saprophytic. It grows on dead and decaying material. But before we proceed, uh, there is a slide to make you understand whatever we grow in our lab or at our home, it is like growing an organism. Mushroom is also an organism. So if we want to grow, cultivate an organism, we need to understand what it takes, how, what kind of food it consumes. Like if we want to rear a buffalo or a cat, we need to understand what their diet is. So in order to grow mushrooms, we need to know what kind of diet they consume, what is their food, what kind of nutrients they need, and how they can be provided in the mushroom lab or culture room of mushrooms. So I'll be starting with some terms which I'll be using during this presentation. First is compost. Compost is a partially decayed organic material which is having nutrients necessary for the growth of button mushrooms. So compost is partially degraded material. Already some macromolecules are degraded into simpler molecules so that fungus can consume them easily. Right. So mushroom compost should be partially degraded in case of button mushrooms. Some other mushrooms like oyster mushroom is there, like paddy straw mushroom is there. So in those cases, they are mostly wood rotting fungi. Like oyster mushroom is a wood rotting fungi. So in order to grow the oyster mushroom, we need to dip the paddy straw or wheat straw, whatever organic matter is there. We need to dip it in water. We need to boil it or sterilize it, and that can be used as a substrate directly without doing composting. But for button mushroom, which is a saprophytic mushroom, we need to first partially degrade the material. But for oyster mushroom, we can directly grow it on the substrate by wetting it, and by sterilizing it, we can directly use the substrate like paddy straw. But in case of button mushroom, we need to make the compost. So I think compost part is clear. The second thing is casing soil. So this slide, when I prepared this slide, it was meant for a presentation which I was to give to farmers in Punjab, one of the villages nearby. So for them, I put another image below in which there is some dish, and above that dish it is garnished by the coriander leaves. So making the farmers understand, I explain to them, just like on a dish, on the top of a dish you may sprinkle some cut coriander. Likewise, once compost is ready, you have introduced the seeds of button mushrooms in them, and the seeds, they give rise to the mycelium. Then above that material, in each bag where we are growing mushrooms, we need to put a small layer of a material which is called as casing materials or casing soil. So I'll be using one term compost, one term casing soil. I think these terms are clear now. The next term which I will use is the spawn. Spawn, you can say this is as good as the seed of mushrooms. Exactly, mushrooms don't have seeds. They are either grown from spores or from the mycelium. So what we do to make a grain spawn, we usually dip wheat or other grains into water. We add some additional materials like calcium, etc., sterilize it, and then put some fungus into it. Fungus colonizes the grains, and these grains are used as the seed of fungus, uh, of button mushrooms. And we use these seeds, these wheat seeds with white threads on it, which is fungal mycelium. We use them as mushroom seeds, right? So spawn is nothing but mushroom seeds, you can say, right? So in order to prepare compost, which is the next step, compost is the material which is a food, you can say, in layman's term. This is the food which the mushroom will be utilizing. So to make compost from most common material, which is wheat straw nowadays, so there are two methods. One is traditional method, long method, and now we are using the method which is short method, right? So quality of compost is very, very critical. If compost is not of a good quality, mushroom yield will be very less. There can be contamination in mushroom bags, or mushrooms might get diseases. So quality of compost should be very, very good. There are two methods, long method and short method. Obviously, long method, as the name indicates, it takes more time. Shorter method needs less time. But what additional we do in short method, we we do pasteurization. We partially sterilize. We sterilize the material, the compost. So the yield is high in case of short method, but we need more mechanization. We need steaming chamber. We need pasteurization chamber, steamer boiler. We need in long method. We need more time, right? And because of microbial activity, there is heat inside the piles of the. I'll show you later. In long method, composting is done on a cement floor, uh, pakka floor, right? Because if more soil is there, it is likely to be contaminated. So we do it on a cemented floor mostly. That's what I'm calling pakka floor. So this is usually done under a roof, because otherwise rain and other dust particles, they can come. It is open from all the sides, but there should be one roof above it. Many formulae are available for the recipe of this this compost. Traditionally, horse manure was being used. Then people started putting chicken manure, because chicken manure is a good source of nitrogen. So people tried different recipes. We usually use gypsum, urea, some other fertilizers which give nutrients, which add nutrients into the recipe of compost. So this is the basic recipe which is used in northern parts of India. Although this is not the only recipe, there are many other recipes also, right? So we use wheat straw, wheat bran, urea, gypsum. Many other recipes are available. So short method has two stages: outdoor composting and steam pasteurization. And long method is done outside only. This is how we first wet the wheat straw. We make it stacks. We add other ingredients like gypsum or chicken manure or urea or DAP or all, whatever your recipe suggests. There are so many recipes, you need to follow one, right? Different people, different laboratories, they give different recipes. The thing is, everybody cooking maybe a dish, maybe any dish, have different recipe, but the end point is same. Everybody have a different style to cook rajma. Likewise, recipe can be different, but this compost, that should be partially degraded. It should be free from microbes. It should have essential nutrients like nitrogen, like calcium, like sulfur, phosphorus. It should have nutrients which are required for the growth of fungi. So after mixing the ingredients, piles are made and frequent turnings are given, and compost piles, they are broken, turnings are given, again piles are made in the long method. Whereas in short method, after some time we do pasteurization, and for pasteurization we have steamers, boilers with us, and a pasteurization chamber. So in short method, which is now all mushroom growers use, rarely nowadays the long method is used. All of the people, all of the mushroom growers, they use short method only, because it is less time intensive, it is less labor intensive, we need less labor. Within shorter duration we can grow mushrooms, and the quality of compost is good. Unnecessary microbes are not there. So in first phase, barley, chicken manure, they are added to wheat straw, and uh, wheat bran is added. Then all recommended fertilizers are added, stacks are made, turnings are given. Turning means you break the pile and then again mix the ingredients. Inside comes outside, becomes outside, and outside becomes inside of the new pile. So turnings are given, and then the next stage is steam pasteurization. In this pasteurization stage, temperature is raised with the help of steam, and harmful nematodes, mites, competitive molds, they are killed. But only heat-loving bacteria, they love to live in such environment, and they consume the free ammonia, they consume other materials and make protein-rich material biomass, which is used by mushroom, which they eat, they consume. So then at last, fresh air is introduced, steam supply is cut off, we let it come to a lower temperature, and when it comes to 25 degrees Celsius within 4 5 days, 6 days say, then we fill them in the bag. So short method, long method, this slide is about the comparison of these two methods. Short method is less polluting for the environment. Long method is more polluting because more things leach out from the compost, and it prepares within a shorter span. Although it requires more electricity, it requires less labor, and the yield is high in case of short method because contamination is less. Mushrooms are healthy, compost is healthy. So generally for a larger scale we always recommend shorter method. Nowadays long method is not generally used. So once the compost is ready, we fill it into the mushroom bags, polythene bags. We fill the bags. These each bag usually contains around 10 to 12 kg of this compost. The final compost should not have any ammonical smell. It should not be too wet. Like if you squeeze in a hand, water drops should not come out fast. It should not be very dry. It should not be very wet. Right? Your hand should be free from water, but it should contain some moisture in it. Once it is ready, free from any foul smell, we fill the bags and we add mushroom seeds, spawn into it. That's why this is seeding. So good quality compost around 25 degrees Celsius temperature is used for mixing the spawn, mixing the seeds into it. Filling of spawn is generally done in polythene bags. Earlier, wooden trays were used, but nowadays polythene bags are more commonly used. And then we close them and keep it in the culture room. Once all of your content of the bag that contains fine threads over it all around, it means that that net-like structure mycelium has grown all over. So after that we put casing material over it. Casing material is required for fruitification. From for that buttons to come out, we require casing material. Earlier, casing material was subsoils or organic matter rich soils. Sometimes peat was used. Peat is not commonly available in India, but it is most desirable material. So in this region we use that cocoa peat. Sometimes we use that cocoa peat, and we sterilize it and use it as casing material. Many alternatives are there for casing materials. Farmyard manure you can use spent mushroom compost. Once it is mushrooms are grown, and then the waste which is there, we can keep it for 2 years, and then we can sterilize it and use. We can use coir industry waste, other different materials are used, but coco peat is easily available. That's why most of the mushroom growers, they use coco peats based materials. Some use spent mushroom compost, which is sterilized, and then they use it. Quality of casing material should be very good. It should be soft in texture. It should be lightweight, not very heavy, but with high water holding capacity. This is very important. It should absorb the water, because whenever we water the mushroom bags, we water them from the top. And on the top, there is a layer of casing material. It should hold water. That's why moisture will be retained, because if it is high water of high water holding capacity, it will retain moisture. It should have high porosity, and it should be deficient in some nutrients. Otherwise, there also thread-like structures will come up, but buttoning will not start. It should be of neutral pH reaction. It should not be very, very alkaline or very acidic in nature. It should not have many ions in it, so it should have low conductivity. These are the characteristics of a good quality casing materials. So once we have casing material is ready, it is pasteurized. Casing material should be free from microbes. So when it is pasteurized, and after pasteurization it is cooled again, and then a layer is put over each bag. So this treatment is called as casing treatment. And then after casing application, these bags are kept in the culture room. When we apply casing material should not have any lumps in it, but there can be mounts and valleys over the top layer of mushroom bag. But um, these bags, once casing is applied, these bags are kept open, not closed. Earlier they are closed, and when the mycelium grows into it, it becomes all wide. Then we open it, we put some casing material, and we keep them open from the top, right? So once mushrooms are ready, how to harvest, which is the appropriate stage to harvest? There are different stages of mushrooms. So stage 2, stage 3, stage 1, people usually harvest in these stages. 4, 5 are not good, but the ideal stage is stage 2. This is most palatable stage. Stage 3 is also good for consumption. Stage 1 is also good. Stage 2 is ideal, most delicate, known for its like mouthfeel. Best conditions for fruiting is 15 to 17 degrees Celsius temperature. Relative humidity should be more than 85%, and carbon dioxide concentration should be around 800 to 100,000 ppm. Although it can vary for by strain to strain, what kind of mushroom seed we are using, which strain it is. So mostly these conditions we keep inside a growing room. When we harvest, we need to rotate it either clockwise or anti-clockwise. So mushrooms are harvested by holding them between the thumb and the finger. We twist it, because if we twist them, the fine threads attached to the base of mushroom, the stalk of mushrooms, they are broken. But if we pull them out, they also come out. So this is not a good practice. We need to rotate them before pulling out, right? So mushrooms, after we pull them out by rotating, we need to cut the edge with a sharp knife, and then we clean it. Some people although use sodium hypochlorite to dip them, because it is used for sterilization as well, and it retains color, but it is always not recommended. So this is how mushrooms are harvested. And mushroom bags, they require repeated watering. And once we harvest mushroom from one part, that part it looks like depressed. So at that depressed area, that area from where casing material has been removed. We just cut the mushroom, and then sometimes we need to add extra casing material there. And for packing these mushrooms, different materials they come, but the ideal one is with little ventilation. Too much washing, cleaning, if we do, excess of water during packing, and if it is there in the packet, it can spoil mushroom, it can reduce the shelf life of mushroom. This is a small article we published in a magazine. This is oyster mushroom farming for beginners. We published it. Farmers which are interested in oyster mushroom farming, they can read it. Very simple method is given there. It was published in a magazine. So we grow pleurotus mushroom, oyster mushroom also. This is relatively more easy, but people usually don't know about oyster mushroom. That's why oyster is not that popular. This is how we grow oyster mushroom in our lab. Students, they are making the bags ready. This is another oyster, gray oyster mushroom, which is Sajar Kaju, and we grow milky white mushroom also in our lab. And these two images are, although not from our lab, I could not find these images from our lab in my phone, because just one day back I was told to prepare this presentation. Otherwise, we are growing this pink oyster also, and sometimes paddy straw mushroom also in our lab. Thank you. Thank you for a patient hearing. And I think uh, the knowledge you gained through this presentation, you can use this knowledge to start your own enterprise if you want to create your own enterprise for mushroom cultivation. And this is very good enterprise as far as environment is concerned. We mostly use the waste materials like paddy straw, like wheat straw, like other crop residues we can use for, especially for oyster mushroom. We can use so many kind of materials which are like agri waste. We can say they fall into that agri waste category. Paddy straw management is a challenge for Punjab and other regions of India. So if we grow paddy straw mushroom, or we grow, it can be used to grow the oyster mushroom as well. So this is how we can manage agri waste. So this will add one more choice of food into our plates. Some of these pleurotus mushrooms, they are very nutritious. They contain some rare antioxidants also, and this button mushroom is rich in proteins. This is a high protein diet, so it can be used to fight protein and energy malnutrition as well. So I think this is sufficient for the day. If there are any questions, I'll be happy to answer.
Thank you, sir. Dear participants, if you have any doubts and queries related to mushroom farming, can ask to sir directly.
Hello, good evening, sir.
Good evening.
Sir, I have a question that if I want to start a business, then what will be the cost of everything required and how much money I have to invest?
First, if you want to start an enterprise with button mushrooms or oyster mushroom, if you want to grow oyster mushroom, your like initial investment could be as less as maybe 50,000 or 1 lakh only, if you want to grow oyster mushroom at first. If you want to grow button mushroom and you want to grow it during winters, Then you can take this material from outside, the seed, the spawn you can take from outside, and you can have your own facility. If you're using long material, you need to develop a pakka floor with the roof on it. And if you are using short method, you need to make arrangements for a pasteurization chamber, for a boiler for steaming and all, and one refrigeration facility if you want, for storage only. And if you want to do it during the summer season, then you have to control the temperature, because mushroom grows at a lesser temperature, around 15 to 20 degrees Celsius, and the temperature here goes up to 45 degrees in the summers. So you have to develop a controlled temperature facility, and for that you need to invest on a large scale. If you want to start, at least 50 lakhs you need to put into it at large scale, at with all temperature controlled facility, if you want to start. And there are different schemes for the loans from government, and some subsidies are available. You can contact with your local, I don't know if you are from Punjab or not, but you can always coordinate with your state horticultural department. They usually help the farmers to establish such facilities. Different states have different subsidy policies, so you can take help from them.
Uh, sir,
Yes.
someone is asking how we prepare the seed of mushroom.
This is very simple. The most commonly used seed is of grain spawn nature. So for that, we soak wheat, mostly wheat seeds in water, and then we add sometimes calcium into it, and we sterilize it by autoclaving. We put some initial inoculum of fungus into it, and then we let it grow in a temperature controlled incubator. And once the mycelium is growing, it uses wheat grains, and all around wheat grains, white thread like structures, they come out. So it is very easy, not at all difficult. But the autoclaving facility, the laminar airflow cabinet facility is not generally available with farmers, so that's why they usually buy it from a lab. Otherwise, it is very simple. We just need to have a laminar airflow cabinet to inoculate, and one autoclave. Autoclave is for sterilization, and laminar airflow cabinet is to inoculate that grain with the help of some initial inoculum without any contamination in aseptic environments. That's why these small two equipments are used.
Thank you, sir. Sir, Shubham Vaidya is asking what about demand and production of mushroom for enterprise?
Demand and production, I'm not sure about every state and every region. It depends region to region. So I'm not sure which region he's asking, and it is different for all the regions. And demand, as far as mushroom variety is concerned, the most commonly demanded mushroom in the market is button mushroom. Paddy straw mushroom has less demand because its color is not very good. It is grayish in color. One gray oyster, it is very delicate, very like, it is very famous among the people who have tasted it once. Gray oyster is very good to eat, but gray oyster, people don't like gray color. Pink oyster, it is very colorful, but its stalks are little harder, so we need to remove the stalks before preparing it. As far as demand is concerned, most demanded variety in India is white button mushroom.
Sir, Dipanshu Pandey is asking, sir, how to prepare spawn?
That I already told you as an answer of the second to last question.
Okay, sir. So next question we have from Niladri Bhattacharya. Which mushroom should be directly grown from compost or grown on compost?
Uh, See, there are two kinds of mushrooms. I told in the initial part of my presentation, button mushroom is grown on compost because it is saprophytic in nature. It grows on dead and decaying material. We need to partially decay the organic material to make compost, and it grows on compost, right? And we add urea, gypsum, ssp, dap, some other things we add to make to add more nutrients into it. Only function of these things which we add into the composters to increase the nutritive value of compost so that more mushrooms can come out. Otherwise, if we don't use them, the yield will be very, very less, right? But some other mushrooms like wood rotting fungi, this pleurotus, oyster mushroom is basically wood rotting fungi. It has enzymes. The fungus releases enzyme into the base material, which we call substrate, and directly it degrade the substrate. It take nutrients from substrate, so we need not to prepare any compost. It can directly take all the nutrients from wheat straw itself. No need to degrade it in case of this oyster mushroom. So oyster is more simpler. We need not to prepare any compost for oyster. Only wet the organic material, wet organic material is used as a substrate. But for button mushroom and likewise for other mushrooms, we need to wet them, not only wet them, but partially degrade them with the help of microbes. And they more easily they take material nutrients from compost, not from the intact substrate. It should be partially degraded, right?
Sir, any commercially spawned seed material is available in the market?
Yes, spawn is commercially available. In Punjab, many mushroom farms, private farms are giving, but I will recommend not to purchase from private players. It is little costly there. You can contact Punjab Agriculture University Microbiology Department. They are giving, I think, 80 or 90 rupees per kg. They are giving, or you can take it from Directorate of Mushroom Research, Solan. If anybody is listening from Chandigarh or Himachal region, they can purchase it from there. Solan, uh, Chambaghat. There is an institute, Directorate of Mushroom Research. You can purchase from them. I think not more than 100 rupees kg.
Sir, how much is the ROI?
ROI?
Yes, sir. Somebody asked about ROI.
Uh, can you spell it full? ROI? What do you mean by ROI? Can you expand?
Sir, I think return on investment, sir.
What?
Sir, return on investment.
This, I think you can better ask any economist.
Okay, sir. Thank you. One question is there, sir. Having that much of nutrients, why is mushroom not recommended for animals?
Mushroom is not recommended for animals. See, mushroom is a costly commodity. When we were studying mushrooms, when we were students and we were studying mushrooms, so my teacher recommended me one book, and that book was from the travelers, how to uh, how to decide whether a mushroom is poisonous or edible. This book was about that. So I read the book, and in that book it was written that if any mushroom is eaten by the rabbits, then it is safe for human consumption as well. So it indicates that animals also consume, but since it is a costly commodity, it is not cheaper. It is cheaper to feed buffalo with barsim. But if you give buffalo so much of these mushrooms, think how much it will cost. So it is, I think, on economic grounds that we better feed them with the traditional feeds, which are cheaper.
Okay, sir. Last question. What are the most common diseases and pests in mushroom cultivation, and how can they be controlled?
Yes, there are so many pests and so many diseases. What I felt in our laboratory, that most common disease is number one, wet bubble disease. Number two, green mold disease, which is trichoderma. These are most common we faced in our laboratory. And one, fruit fly, like flies are coming, they're tunneling into the mushroom sometimes. So we are facing these. Otherwise, there are many diseases, and this is a different topic. If I want to discuss all the diseases and pests of mushroom, it will take 1 hour at least. So this is a long subject. But the most common here in Punjab, in Jalandhar region, is wet bubble disease, and secondly, green mold, trichoderma.
On behalf of Edu-Mentors Pvt Limited, I would like to express our sincere gratitude to Dr. Ashutosh Sharma, Associate Professor, Faculty of Agriculture Sciences, DAV University, Jalandhar, for delivering such an insightful and inspiring session of sustainable mushroom farming. Sir, your expert guidance on sustainable mushroom cultivation, scientific production techniques, opportunities for entrepreneurship, and the importance of mushroom farming in promoting sustainable agriculture has been immensely valuable. Your practical insights and rich expert experience have greatly enriched the knowledge of all our participants. We are truly thankful for our valuable time, enthusiastic participation, and willingness to share your expertise with us. Your session has motivated our students, researchers, faculty members, and professionals to explore the vast potential of mushroom cultivation as a sustainable and profitable agriculture enterprise. Once again, thank you very much, sir, for your excellent presentation and for being a part of our 21-day international summer school training program on climate resilient agriculture and allied sciences. We have looked forward to welcoming you again in our future academic programs. Thank you, sir, and best wishes for your continued success and contributions to agriculture, education, and research.
Thank you so much. I'm logging out.
Participants, you may leave.
Thank you.